Okay, so next we want to take a quick look at the types of cells. So we mentioned a quick list of the major types of microbes that we're going to be studying. Before we go into any more detail about that, which we will here shortly, I want to just go over some of the broad ways to look at all cell types, not just microbes, but actually all living cells on Earth. So from a broad standpoint, we can look at all life in two categories from a cell perspective, and these are considered prokaryotes and eukaryotes. So first of all, taking a look at this line here, all living things are made up of at least one cell. This is a somewhat of an arbitrary distinction in biology, but a relatively a sound distinction, and that is that life begins at the cell level. That's basically what that's saying. It doesn't say that, obviously, but the idea is that you have to have at least one cell to be considered a living thing, and then many organisms have many more than that, obviously, but that's kind of where life begins. So when we're comparing all life forms on Earth, we really have no choice but to compare them at a cell level, because not everything has multiple cells. Now we get into animals, for example, we can compare tissues and organs and body structures and all that kind of stuff. But from a very, very, very broad view, we have to compare life on a cell-by-cell level, because many organisms, as I've already mentioned, only have one cell. Or I should make that clear if I haven't already. Many organisms are made up of only one cell. Therefore, we have to use that as kind of the fundamental basis of comparison. So prokaryotes, pro stands for first, U stands for true, and the idea is that these were the first cells, the prokaryotes, and then the eukaryotes, they called these the true cells, because as we'll see here in a minute, these are more complex and a little bit more of what we are more familiar with in terms of more complex organisms. So prokaryotes, one thing you want to know about them is that they're always single-celled, and they're relatively simple. And I put that in quotes because there's really no such thing as a simple living organism, but these are relatively simple. And by that I mean that they're simple in comparison to the eukaryotes. So on a comparison basis, they're simpler. So recognize that they're single-celled and relatively simple. Again, the pro stands for first, so they were the first cells to appear on Earth. Now, these lines down here, DNA is not enclosed by a membrane, no cellular organelles. These will make more sense later. Right now, we don't have enough to really go over that, so we'll point these out. I want you to recognize that these are characteristic to the prokaryotes, and recognize that we'll understand these when we get to later chapters. Chapters 4 and 5 will make a little more sense of those two lines there. Eukaryotes, what I want you to recognize about these starting out is that they are relatively complex, again, in comparison to the prokaryotes. And also I want you to recognize that they can actually occur in both single and multicellular forms. So if you see something with the naked eye, a human being, a dog, a tree, any kind of plant, any kind of insect, any kind of animal, even things like algae and pond scum, if you can see it with the naked eye, then these are considered a eukaryote, because it's really only eukaryotes that are multicellular. All prokaryotes are single-celled and thus very, very, very small. So eukaryotes are the only ones to occur in a multicellular form. Now, to clarify this, don't get this confused, eukaryotes can actually be both. So they can actually be single-celled and or multicellular. So if you can see it with the naked eye, it has to be a eukaryote. Prokaryotes never get large enough to see with the naked eye. If it's microscopic, on the other hand, then it could be either a prokaryote or a eukaryote. So eukaryotes can be microscopic and macroscopic, whereas prokaryotes are always microscopic, meaning that they're single-celled. Now, eukaryotes then have the opposite characteristics down here with these two lines, and that they do have DNA enclosed by a membrane, and they do contain cellular organelles. So let's take a quick look at a figure here showing the two. On the left, we have a prokaryote. I like to think of prokaryotes in terms of a house, or I like to think rather both cell types in terms of houses. And I like to think of prokaryotes as being really kind of like a single room apartment, like a studio apartment, or like a log cabin. It's a house, it's a place to live, it's got a structure to it, but the structure is relatively simple in that it's really just got this one enclosure here, one kind of area, one area where everything is enclosed. Think about it like a single room apartment. So you've got kind of the walls around the outside. In a cell, this is a cell membrane. We have a cell wall, and then in some cases we even have an extra layer called the capsule. In this particular example, not all bacteria have that. But to focus here, we're looking at kind of the basic cell, and then inside we really just see kind of this one area where everything takes place. And that's kind of the idea of the quote-unquote simple characteristic of a prokaryotic cell. Kind of like a studio apartment, it's a relatively simple living environment, and everything takes place in that one area. Kind of like a studio apartment, you've got your living room and your kitchen and your bathroom really all kind of in the same area, which is the idea. With the eukaryote, think of it more as like a traditional house. You've got a living room, you've got a bedroom, you've got a bathroom, kitchen, etc. And the idea is that while they both serve as a living environment, the multi-room house is a little bit more organized and more complex, and that's kind of how you can think of a eukaryotic cell. It's got these divisions of space within the cell, these kind of different compartments, so to speak. And we'll get into more detail on what all these different compartments are, but for now I just want you to get a visual and an idea of what we're looking at here. And just to back up here, when it says the DNA is not enclosed by membrane, DNA is enclosed, and then no cellular organelles, and then cellular organelles. The DNA in a eukaryote is found within this big elaborate structure here in the middle of the cell called the nucleus. So DNA is important to a cell. You're probably familiar with the idea of DNA is kind of what makes you who you are and gives you your characteristics. It's true for all living things. And in a eukaryote, that DNA is housed and processed within this special center called the nucleus. In a eukaryote, the DNA, they both have DNA, but the DNA is just kind of floating in the middle of the cell. Not really compartmentalized, not separated, just more or less in there with everything else kind of sitting in the middle of the cell. Okay, and in a eukaryote, it's again separated and kind of compartmentalized. So scientists saw this as a fundamental difference between these two major cell types, and that was really the major distinction that they went by in classifying the two as separate. So prokaryotes they looked at and said they don't have this, and in eukaryotes they do have this, and again they found this as kind of a fundamental difference in all cell types, and so they decided to use that as kind of the primary basis for differentiation. So that's really what it comes down to. So recognize that DNA is kind of the key to all life, especially cells, and how it's housed and organized is therefore like the major differentiating factor in the two. And then the cellular organelles are these additional little compartments, things like the mitochondria. You got things like called Golgi apparatus, rough endoplasmic reticulum, and smooth endoplasmic reticulum, which actually surround the nucleus. These are additional organelles that have extra functions, kind of like your bathroom has a certain function and your kitchen has a certain function. In a eukaryotic cell, these have their own space for their own function. And in a prokaryote, you just don't have that. They can carry out some of the same general functions, but they can't do it on the same level of complexity. And this is partly why we see prokaryotes always being single-celled and relatively simple, rather. And we see many eukaryotes that went on to become multicellular and very complex, elaborate organisms like human beings are. And part of that ability lies in the organization at the cell level. Okay, so that's kind of a major difference between the two there. Now, next I want to talk about some of the prokaryotic organisms. There's only two broad groups of prokaryotic organisms. So these are still relatively broad groupings within a broad cell type here. One you're probably familiar with, bacteria. The other one you probably never heard of, and these are called Archaea. Now, we're not going to talk much about the Archaea. We're going to focus mainly on the bacteria, but I want you to recognize that these are the two major types of prokaryotic organisms. So, as it says here, bacteria will be a major focus of the class. They're the most common type of living organism on Earth. That is true. So, they are very, very, very common all throughout the world, throughout our bodies. As you'll be reading online and learning in class, these microbes, bacteria specifically, make up a big part of our intestinal tract. They make up a big part of our skin, our mucous membranes, like our nasal passages and things like that. So they're not only very common in the environment, but they're also a part of our bodies and part of our tissue and our organs. So for that reason, they're relatively significant. Now they're simple, but they have apparently, according to some of the latest research, have a very significant impact on our bodies as well as the world around us. So a lot to talk about here. In fact, really, chapter 4 is going to focus on the bacteria specifically. So here we're just going to look at a real kind of a broad perspective. So recognize that they are a prokaryote, one of the most common, or really the most common type of living organism on Earth, one that you never really see, but always there. And remember the term ubiquitous. These are ubiquitous. Now, to clarify, we say microorganisms as a whole are ubiquitous. Just to back up here real quick, when we look at this slide, we say microbes are ubiquitous. Remember that microbes are lots of things, right? These are all considered microbes. The truth is, not all of these really are ubiquitous. They're ubiquitous as a whole. It's actually really the bacteria that are the closest to being truly ubiquitous all on their own. Bacteria really truly can be found in almost every location on Earth. It would be an extreme challenge to find a location where they wouldn't be. Absent, you could definitely sterilize something and take steps to remove them, and then obviously they wouldn't be there. But if you weren't doing that and you were just trying to find a place where they wouldn't exist naturally, it'd be very, very difficult to find a place where you wouldn't find any bacteria. Almost impossible. And we'll demonstrate this in the lab. For those of you taking the lab, we'll do what's called a ubiquity lab, where we actually culture for bacteria and another type called fungi, which we'll talk about, and we'll find them everywhere we look. I maybe have seen one culture dish in the 5 years I've done this that didn't grow anything. So one of the things that bacteria will typically do is when they get a nice nutrient-dense location, they will start to grow into colonies, or in some cases what they call a biofilm, more of like an advanced colony. Now, in this case, they actually can become visible to the naked eye. So individually, on a single cell level, which you're looking up here, these are all individual bacterial cells. By the way, that's a real image. That's not a computer animation. It looks like a computer animation, but it's actually a very, very high-def microscopic... Oops, I keep doing that, sorry. It's a very high-definition what's called a scanning electron microscope image. So these are individual bacterial cells here. Don't have a picture of a colony. I'm sorry, I should have one here. Let me find one here for you. So here we see some colonies growing on petri dishes. All of these represent, let's take a look at one of these here. Here's a colony on a petri dish, 4 millimeters, pretty small. Each one of these little circles is a colony, and each one represents probably millions, if not hundreds of millions of bacteria. So in a colony approximately 4 millimeters in diameter, you could easily have several hundred million bacteria in that one little area there. So they are extremely small, and individually or even in small groups would be completely invisible to the naked eye. But if they get a nice nutrient-dense location to grow, then they can become visible in a huge mass. It's like kind of like the being able to see a large city from kind of like the edge of space, or like they say they can see the Great Wall of China from space. So something big enough you can see it from a long distance away. Individually, they're too small to be seen. This is actually the head of a pin. This is a high definition microscope image of the head of a pin, and here you can see lots and lots of individual bacteria, a couple clusters of bacteria there scattered about on the tip of this pin here. So that kind of gives you a visual there. We'll talk about biofilms, and we will grow colonies in the lab for those of you taking it, so we'll get familiar with those. A little bit redundant here, as it says bacteria are found everywhere. We'll talk about more details of bacteria here. So for now, recognize some of the facts that I pointed out there on the last slide. Next, let's talk about the Archaea. I'm not going to talk about these much after this, so I'm just going to mention them real quickly and then move on. This is the other type of prokaryote, so you want to recognize that. You also want to recognize that they're generally considered quote-unquote extremophiles, meaning that they love extreme environments. And they got this designation because they tend to find this type of organism in locations where other organisms wouldn't be found. So this would be an example where you might not find bacteria in certain locations, but you would find Archaea. And for that reason, they call them extremophiles. So There's a number of examples. You can find them in really acidic environments. You can find them in extremely hot environments. This is a hot vent in Yellowstone National Park, where some of the Earth's magma, lava comes close to the surface, heats up some of the water pools near the surface there, and that creates a very extreme kind of hostile environment. And this is a location where you can find all sorts of little microscopic archaea growing and thriving in these locations here. Now, they're not all considered extreme, but because so many of them are, that designation was given to them. They're kind of similar to the bacteria in that they are prokaryote, but they are different at the same time. So we don't really want to get into that because that's actually more of an advanced biology topic in terms of the archaea. Really, archaea are interesting, but they're not usually a topic for an introductory microbiology class like this one. And that's because they tend to be niche. They tend to have a very specific type of location and environment in which they grow and live. And as a result, we don't really want to focus on the outliers. We want to focus more on the norm and what's more common, especially what's medically relevant and environmentally relevant in most cases. So for that reason, we won't really talk about these much more after this. So I just want you to recognize some of those facts I just pointed out there, and we will move on from these here.